Yes, okay. It's my pleasure to introduce once again Dominik Wodarz, who is coming to visit us via Zoom from UC Irvine. So it's not the first time that Dominik gives a seminar, but a few things have changed since last time he was here. For starters, that he's actually not here. Clearly, something has happened in the last 12 plus months, which also brings me to the point that as opposed to most of us at the Math Onco, where we actually sort of exclusively work on Math Onco, you actually have a few papers of lately on COVID-19, right?
Yes, we've done a few papers.
But today is going to be a talk about homeostasis and cancer, which I think is touches my favorite topics. So as it was last time, you still had a bachelor's in biology from Imperial College London, a DPhil from zoology in zoology from the University of Oxford, and last time you were here, you were a professor at the Department of Ecology and Evolutionary Biology, but now you're a professor at the Department of Population Health and Disease Prevention, still at UCI. So you can tell us what that change represents to your research or to your focus. And other than that, please, Dominik, take the stage is yours.
Okay, thank you very much for the invitation and for the introduction. Yeah, the department change doesn't reflect any change in my research, just reflects a change in department, and I'll leave it at that. So today I'm going to talk about the dynamics of feedback regulation in healthy tissue and tumors, and that's obviously a very sort of large topic with lots of things, and we're going to focus on a few key areas. So I'm going to start giving an overview of the types of mathematical models we even others as well have been considering with feedback, tissue hierarchy. Then I'm going to spend some time talking about how feedback control might work in spatially structured populations. So most of the work that's related has been done with ODEs that assume perfect mixing. So we'll look at what the spatial structure do. And then the final part is we'll look at feedback control and how that can influence resistance against chemotherapy in bladder cancer. Okay. Well, why does it not work? Oh, there you go. That's how I change slides. Okay, so healthy tissue architecture, we start simple. We start with some very basic principles. You've got stem cells. Stem cells can self-renew. They maintain the tissue. They can differentiate. When they differentiate, they give rise to transit amplifying cells that can, to some degree, self-renew, but cannot maintain the tissue by themselves. And they then differentiate and give rise to differentiated cells. And that's a sort of very caricature representation of what's going on. What's going on is obviously much more complicated, but this is sort of the frame of mind that we have. So when we talk about maintenance of homeostasis and thinking about how these stem cells work and how to model them, there are various ways in which one can do that. And there's some uncertainty about how important the different processes are. So one process is called the asymmetric cell division. I'm sure everybody knows what that is. You have a stem cell. When it divides, it gives rise to one more stem cell and one differentiated cell. I hope you can see my mouse pointer since I'm pointing with it. So it's asymmetric. Sorry?
We can see it.
Okay, good, thank you. So basically the two donor cells are different in this way. You maintain the stem cell population and you also give rise to a differentiated cell. And that's been shown in Drosophila to be quite important, and I think in higher organisms as well. But there's a different way of thinking, a different model where you have only symmetric divisions, but it's probabilistic, and it has kind of the same effect on average. So when a stem cell divides, the stem cell with a probability P gives rise to two new stem cells. That's a self-renewing division. With a probability 1 minus P, the opposite probability, a stem cell gives rise to two differentiated cells. And in the process, the stem cell is obviously lost, and you have two differentiated cells. I'm making this even simpler than before by ignoring the transit amplifying compartment. These are just differentiating divisions, whatever differentiated cells, eventually they'll be terminally differentiated. And so the thing is that on average, these two have to balance each other out so that you maintain the stem cell population and you also produce differentiated cells. So this mechanism is supported by quite a large number of data in mammalian tissue. So I think asymmetric cell divisions do happen. There's evidence for that, but there's quite a bit of evidence that this stochastic symmetric cell division model is quite important in mammalian tissues, that on average tissue is maintained, but when the stem cell divides, gives rise to either two stem cells or two differentiating cells. So that's the probabilistic model, and that's sort of what we've been concentrating on in a number of modeling studies. So this can be described by a relatively simple mathematical model that's been also used by a variety of people. It's very straightforward. This is the simplest way in which you can describe it. I like describing things in the simplest way possible because you can better understand the dynamics and the consequences, and then one can always build complexity further on. So we're ignoring the transit amplifying compartment, which is just kind of, we can assume that the stem cell compartment comprises both the stem cells and the transit amplifying cell. It's a simplification. But essentially, the cell in this model divides with the rate R. When that division occurs with a probability P, you give two daughter stem cells. With a probability 1 minus P, you give two differentiated cells, and the differentiated cells die with the rate delta. Stem cells in this particular model are not assumed to die. Stem cells do disappear, but they disappear by differentiating out. So in this model, it's not a very realistic model. There are basically three outcomes. If the probability of self-renewal is less than 0.5, the population cannot maintain itself, population goes extinct. If the self-renewal probability is bigger than 0.5, you get exponential growth, population blows up. A constant population, which is homeostasis, is only maintained if P is exactly 0.5, which means that half of the division has to be self-renewal and half of the division have to be differentiating divisions. And so that's not realistic, but that's the basis with which we start. So in reality, what happens if you, on top of this process, you can put regulatory mechanisms, feedback mechanisms, control mechanisms, where cells secrete certain factors, and those factors can influence the behavior of the stem cells and the transit amplifying cells and the differentiated cells, such as negative feedbacks. When there are too many cells, it stops certain processes, and so on. And so this has been looked at in the simplest, in this sort of simplest framework. And particularly two feedback loops have been sort of identified experimentally in the olfactory epithelium, and they are thought to contribute to homeostasis, and the experiments support that in some way. And these are feedback factors that are secreted from the differentiated cells. So differentiated cells produce factors, and these factors can block the probability of self-renewal. So if there are too many differentiated cells, you get less self-renewal and more differentiation. And the differentiated cell also produce factors that block the divisions themselves, so the cells divide less. And so you can plug that into the model by making the parameters P, the probability of self-renewal, and R, the rate of division, dependent on the differentiated cell population, which is the feedback essentially. And if you do that, the system converges to an equilibrium if your basic probability of self-renewal is bigger than 0.5, because then the cells first start growing, and then that growth gets shut down through negative feedback, and you converge to an equilibrium where the population size is influenced by the presence of those feedback factors, by the feedback strength, which is the parameter H here, and also K over here. So this was in this paper up here, this was it was said that these feedback mechanisms can indeed maintain constant cell numbers and can maintain homeostasis. So they're experimentally identified factors that do these things in the olfactory epithelium, and they're supposed to play a big part in maintaining homeostasis there. So in this model, the feedback on the division rate on R actually does not contribute to homeostasis. It's just there to, it influences the dynamics, but it doesn't make the equilibrium happen. What makes the equilibrium happen and what is responsible for homeostasis is the division rate on the probability of self-renewal. And it works in the following way. If you've got too many cells, so too many differentiated cells, then you block self-renewal of stem cells. That results in more differentiation, and differentiating out results in death. So reduce the population, and the whole thing goes to an equilibrium. So in further considerations, for now, I'm going to get rid of the feedback loop on R in this model. And I'm going to concentrate on the feedback loop on P, on the probability of self-renewal. And again, this is a simplified model. There are many more feedback factors that can happen there. People have worked on this. I'm not going into those details right now, because I want to concentrate on this particular feedback loop, because this particular feedback loop on the probability of self-renewal has been shown experimentally, and it's been argued that this is crucial for maintenance of homeostasis. So we concentrate only a model with only feedback on P. So what we wanted to know then is what happened if you put these same assumptions that I just outlined, and we put that into a spatial model in a population of spatially structured cell growth. And to do so, we considered an agent-based model, kind of a hybrid model, where we have two layers. One layer is a stochastic agent-based model that tracks the location of individual cells. So you can have stem cells, which are shown in blue here, differentiated cells in yellow. And when a cell reproduces, you can assume that the offspring of the cell is placed into one of the nearest neighboring spots in a 2D space over here. And we assume that the differentiated cells secrete the feedback factor that inhibits self-renewal. And so every time there's a differentiated cell in a certain location, there's a parallel grid where we model the feedback factors. And the feedback factors are modeled in a kind of very simplistic way by having a patch model, where in each patch you have an ODE that describes the rate of feedback factor production from the underlying cell, the decay rate, and the migration movement of the feedback factors to nearest neighboring spots. So it kind of mimics diffusion outward over here. So that's, and then the cell fate decisions back here in the agent-based model, like the probability of self-renewal of the stem cell is determined by how much feedback factor there is in the in that particular location.
Can I ask a quick question, Dominik?
Yes.
So feedback factor is a very generalized term. Is there something specific one could point to biologically that would give us a little bit more context there?
Yeah, I mean these are factors that are secreted from cells that can belong to the TNF family of factors. They seem to reduce the probability of self-renewal. I think GDF is one of them. So there are a variety of them that have been identified experimentally. I have to look back at the paper to give you exact list of candidates that have been pointed out for the olfactory epithelium, but yeah.
I thought it'd be useful to have a specific example for people to think about. So that was good.
Yeah, so, okay. So that's those factors, and they work according to this patch model deterministically over there. So that's the assumptions of this model. We can add cell migration to the whole thing to make it more or less mixed. So if you assume that cells can migrate and you have a very large migration rate, then this model will converge to some more mixed, more mixed properties. So that's what I'm writing here in purple. For fast cell migration rate and fast movement of feedback factors across space, the dynamics will approximate a well-mixed system. And you can do ODEs. You can write down corresponding ODEs, and that's the first thing that I want to show and do. So let's assume that everything moves really fast, then you can describe the system by this set of ODEs where you have stem cells, you've got differentiated cells, and now you've got also a population of secreted feedback mediators. And the difference compared to the previous model, well, one difference, I'm including the possibility of cell death in the stem cells. It's not very important, it's just written here. But importantly, we have a carrying capacity. Now, we have a finite space in the system because we consider the agent-based model considers a grid, and grid has so many spots, and that's the carrying capacity. That's the maximum number of cells that can be there in the system. So that's an important advantage, an important difference. The new population of secreted feedback mediators that are produced at a given rate by differentiated cells and die. And another difference is in the feedback term over here as well. So in the previous model, it was just simply P0 divided by 1 plus H times Z. But since Z is a set concentration in place, if your grid is larger, then per location, your concentration of the feedback factor will be smaller. So you've got to divide that by K as well. And so this model, if you run the agent-based model that I showed you previously and assume that cells migrate very fast, feedback factor move very fast, it converges to the equilibrium predicted by the ODE, which is shown by those black lines here. So that it converges to this set of ODEs that I've just written.
But you've just removed the entire spatial feature by making it move very fast, right?
Yes. So this is essentially a non-spatial system to start with.
Yeah. But there is spatial structure there if you don't allow it to move very fast. And I guess a lot of stem cells might be niche driven, and that niche might be very localized. And so moving very fast isn't going to help you.
Correct. But I wanted to start with this because I wanted to see the properties of an underlying mixed system.
Well, I understand, but I'm just saying,
yeah.
Are you going to talk about that at all?
Oh, yeah.
Yeah, okay. Good.
Yeah, absolutely. I just want to show this because the ODEs are actually different from the previous ODEs that I've shown you, and that results in some very important differences in properties, I think. One difference in properties you can see here is in the simulation, these are just the stem cells. If your system size, the carrying capacity is 10,000, maximally 10,000 cells, you go to this equilibrium, the blue here. If you have a higher system size, higher carrying capacity, you go to higher number of stem cells. So we have this negative feedback that in the previous ODE gave you complete homeostasis in numbers. In this ODE, it does not do that. The feedback mechanism does not give you homeostasis when it comes to the absolute number of cells, because you can see if you increase the system size, the number of cells grows to a larger size despite the presence of feedback. And that's shown here on the right on the graph where we just have the number of cells at equilibrium, and we plot that against the carrying capacity of the system. This is how big the space is essentially, and the bigger the space, the bigger the number of cells that you get at equilibrium. And so if you've got an infinite space, your K is infinite, you have an infinite number of cells in the system. And so from this, that's why I went to the mixed system first, because we get some interesting insights, is that the negative feedback on the self-renewal in this model can actually not keep total numbers constant. It cannot, it does not lead to true homeostasis. You need the carrying capacity to keep your number of cells limited. So negative feedback, it can keep the density of the cells constant, but not the total numbers. And so I think that's an important point that needs to be made, that in this context, in this model here, the negative feedback on the self-renewal probability does not really maintain homeostasis.
But your feedback is dependent on K though.
Yes. Because
right.
Everything diffuses across the space. So if you've got a huge space and you've got a certain amount of feedback and it's going to diffuse, it's going to sort of be less concentrated per patch, per spot.
Fair, but it feels a little bit skewed because of the way you're modeling space right now. But it'll be interesting to see what happens when you more explicitly consider the space without mixing it so well.
Yes, which is right here. Getting going on that right now. So this is a similar simulation, but now we do spatial dynamics. So with respect to cell reproduction, we assume that reproduction occurs only to nearest neighboring spots, so extremely spatially restricted. You can only put your offspring to the nearest neighbors, and there's limited diffusion, limited movement of the feedback factors from the cell of origin. And this is a graph that shows kind of the same thing that I showed you in the mixed system. We're plotting now stem cells and differentiated cells, and we're comparing a small system size with carrying capacity 10,000 to a larger system size with a carrying capacity of 40,000. And we can see that the same results happen in space, completely carries over, that you increase your carrying capacity, your number of cells goes up. And again, if the bigger your system size, the bigger the number of cells. So again, we say that in this system, now with spatial restriction, it's not possible for homeostasis to be maintained by this feedback mechanism. But other interesting things emerge in this system. So this is a simulation where feedback is relatively strong. So you've got strong negative feedback inhibition here, and what you see here is that spatial patterns emerge, and you get some sort of a clumped distribution of cells across the space. And
what do the colors indicate in this spatial distribution? Yellow and...
Let me just, 1 second, something popped up on the screen here. I want to just put that like this. Okay, so the yellow, so the blue cells is up here, the light blue cells are the differentiated cells. The yellow cells, hang on, I got that wrong. The light blue cells are the stem cells. The yellow cells are the differentiated cells, and the dark blue spots are empty spots. There is nothing there. Okay, so you get this clumped distribution, and each clump is a collection of stem cells surrounded by their differentiated cells, and these are separated from other clumps by empty space. And you can see the corresponding time series here. Stem cells go to an equilibrium, differentiated cells go to an equilibrium. So what we're saying, if you've got strong negative feedback inhibition, although this does not contain or maintain homeostasis in the strict sense, as I just mentioned, it doesn't control the cell numbers, it can give rise to these structures. And these structures, I don't know how to interpret them. I mean, one way to interpret them is that these could be niches, stem cell niches, and that this feedback somehow contributes to the formation of the stem cell niches, because it sort of results in this organization of the system into discrete patches that contain a few stem cells and differentiated cells.
Does it correspond to any sort of biologically relevant tissue structure? And I think it's kind of getting back to Sandy's comment about niches, because again, the niches is not just a clump of stem cells. Usually it's a kind of some element of microenvironment that controls whether the cell can exist in a stem cell phenotypic state versus differentiated state. And I'm not aware of any kind of tissue examples where you have really kind of clumps of stem cells and in the periphery differentiated cells. It's usually few stem cells that are only stem cells and certain spatial environment dictated by interaction with extracellular metrics or other cell types, and then as long as they lose that, they differentiate.
Yeah, exactly. I mean, this is not meant to describe a realistic niche or a tissue in its entirety. We're just, the question we're asking is, what can such a feedback loop do? Just that feedback loop. Okay, of course, there's extracellular matrix. There are all sorts of other things that you have to put there in space. But if you put all of that in, we lose sort of sight of the simple question we're asking, which is what does this feedback loop do in space compared to no space? So once we figure that out, of course, we can put in, if you want to describe a particular tissue or a particular tissue structure, we'll have to put in a lot more complexity. But before we get to the complexity, we've got to kind of understand the basics, and that's getting towards the basics, asking what does that feedback loop do in a spatial setting compared to a non-spatial setting. Okay, and that's
so
what we
basically the deeper question, can just the existence of feedback loop explain these things. I remember I had like this discussion with Bob Weinberg where he basically making an argument based on the feedback loop, and my point was if you have that, you will get the patches of stem cells, and this is not what you're seeing in tissues.
Right, you get patches of stem cells, and I mean this might have some relevance for something. I'm not saying it explains the niche. I'm saying it is different. It does something, right? This is what it does, and then it's talking to clinicians and lab experimentalists like you to kind of figure out what does it mean and how do we interpret this and what do we need to add in order to then sort of move into a more realistic setting. Yeah. Okay, so that's that. And so then the next thing we did is weaker negative feedback. And you can see that with weaker negative feedback, these structures disappear, and also the system gets run over by stem cells. So you can see in the time series that the number of stem cells is much dominant compared to the number of differentiated cells. So this represents some sort of pathological case where things are going the wrong way. Maybe that's something that can contribute to pathogenesis. So this negative feedback on stem cell self-renewal can result in these clumps with more realistic stem cell fractions. Let's put it that way. So we looked at how this clumpiness of the system depends on the strength of the negative feedback. So we just measured the clumpiness of the system by taking squares, looking how many cells there are. And if you take the variance of the cells and divide it by the mean to take that ratio, if that's much bigger than 1, then you have a clumped system. If that's much less than 1, you have a uniform system. And you can see that there's a fairly sharp transition as you go from with strong negative feedback to weak negative feedback. You go from a clump to a uniform distribution. So I wanted to know how do these things depend on parameters? And this is actually important from coming back to the question, what does space do over here? So this is a graph where we ran the simulation many times. Each dot represents an outcome. Yellow is extinction. Blue is a uniform distribution, and red is sort of a persistence in the form of a clump distribution of these patches. And so we varied the strength of negative feedback, H, over here. We always vary that, and then we vary another parameter, which in this case is the rate of feedback diffusion, or the decay rate of the feedback factors here. And the interesting part here is that the parameter region where you get this clumped existence is actually most pronounced if the feedback factors diffuse very fast across the space. If they're slower and it's more, the system is more spatial, then it almost goes directly from extinction, if you weaken the feedback loop, towards the uniform persistence that's dominated by stem cells. So if we've got a really truly spatial system where the feedback factors diffuse very slowly, you almost don't get this clumped outcome at all. You go directly from extinction to this pathological uniform distribution. And the same can be seen here. These are the decay rate of the feedback factors. The longer they live, the slower the decay. Kind of the more widespread these factors become. Again, if they decay very slowly, you've got a significant range of parameter values where you get this clump distribution. But if they decay faster, you again transition pretty much from an extinction to the pathological uniform distribution. So this again casts doubt on the notion that this feedback loop can actually give you homeostasis. Okay, these are other parameters. We looked at the production rate of the feedback mediators, death rate of the cells, probability of self-renewal and division, and well, they don't change all that much in the outcome. So we really think that it's these two parameters that determine how broad this parameter region of the clumped persistence is, and that the more spatial it is, the less pronounced it is. So basically, what we think is that this negative feedback loop on the probability of stem cell self-renewal cannot really maintain homeostasis well at all in a spatial setting, which is very stark contrast to the original ODE model where that was considered. So then we looked at a different feedback regulation, and one that is actually inherently spatial. And that caught my eye in this paper here, and where people looked at the airway epithelium in mice, where you have stem cells, transit amplifying cells, terminal differentiated cells, and they identified a feed-forward mechanism where you have stem cells, and the stem cells produce a factor, which is a notch ligand, and it influences the transit amplifying cells. In the presence of this feed-forward factor, the transit amplifying cells will self-renew and maintain themselves, but if they don't get enough of this signal, they will go and terminally differentiate. And so I found that very interesting, and I wanted to see what that does, because it is inherently spatial. Why is it inherently spatial? Because if you've got the stem cells here, as your transit amplifying cells get further and further away from the stem cell, the feed-forward signal from the stem cell becomes less and less in concentration. And so the probability that the transit amplifying cells self-renew becomes lower, and the probability that they go into terminal differentiation becomes higher. And so this is what we wanted to model then. So we expanded the model complexity a little bit by including transit amplifying cells. These can also self-renew or differentiate according to the same principles as stem cells. Self-renewal mostly happen when there's the feed-forward signal present. It gets reduced in a dose-dependent way. The less of the signal there is, the more the probability that you differentiate out. So in some sense, the stem cells kind of maintain their own niche here by maintaining this transit amplifying population when they are in the vicinity of the stem cells, but they become differentiated when they're located a little bit further away from the stem cells. And so we wanted to see what does that do what is the effect on homeostasis. And so we started with an artificial system, very simple system as I like doing, and we assume no death of stem cells and no death of transit amplifying cells. And if you have that system, then you actually get a self-limited growth, and you get one blob growing, and the total number of cells will stop at some point, and this is independent of system size. So in this system, you do get true homeostasis. And so the cell numbers are kept constant, and this is brought about by competition between stem cells and transit amplifying cells in this system. So this is a completely artificial system, but we'll get some insights from it. We can see here that you've got the blue is the stem cells, the green is the transit amplifying cells, the yellow is the differentiated cells. The differentiated cells only pop up when they're far away from the stem cells, but the transit amplifying cells surround the stem cells and block their division, so they can't really grow out further, and that's what limits the size of this blob, and that's what makes it independent of system size. Now, as I said, this is an artificial system. Now, if you include stem cell death and transit amplifying cell death, this competition can go in one of two ways. If the stem cells are kind of weaker replicators than the transit amplifying cells, and the system goes extinct because the stem cells will be pushed away by the transit amplifying cells and they will dwindle to extinction, or the stem cells can kind of make their way through this green blob of transit amplifying cells whenever they die and push out and push the whole blob out, and it keeps growing, and homeostasis is lost. So this feed-forward mechanism alone, again, cannot result in homeostasis. But then an interesting point here that we then found is if you can combine the feed-forward loop that I just showed you with a feedback loop on negative feedback on self-renewal that I discussed before, and now you do get true homeostasis in cell numbers. So you can look at the plot on the left here, the number of cells versus time. The purple and the blue are the stem cells. The purple is a small system size with carrying capacity 10,000. The blue has a larger system size with 22,500 cells maximally. And so they hover around the same levels. And the fuzz around the lines are the standard errors, because these are multiple simulations across many runs. Differentiated cells as well, they stay at a relatively constant level. And so here's a spatial picture that's typical during that equilibrium phase. You get a blob that has some stem cells, has more transit amplifying cells, and then differentiated cells surrounding that. I didn't plot the number of transit amplifying cells to keep the graph a little bit simpler. But the point here is that in this model, if you compare the feed forward with a feedback loop, you get true homeostasis. You get the same number of cells regardless of how big your system size is. And so this, I think what emerges from this is that if you combine different feedback loops in a complex way, you can get true homeostasis, but this requires spatial components. We have not been able to find that in an ODE in a non-spatial model. So what this argues for us is that these spatial dynamics that we're modeling here are a crucial component that contributes to the maintenance of homeostasis. So we've not found that in non-spatial models, and as I said, this is just one example. You combine these two feedback loops, you find now it works. And I'm sure there are other examples of feedback loop combinations that can be spatial in nature where the same things happen. We just described one mechanism that can lead to that. But the takeaway is that the spatial components are probably important to have the true homeostasis, that they always maintain a constant number of cells. And again, these are simplified models, but this is where we're sort of getting to. So the conclusions of this part is that the negative feedback on stem cell self-renewal alone cannot maintain homeostasis, doesn't maintain total cell numbers. More space will result in more cells, despite the negative feedback. What it can do in the spatial system, give rise to some clumped spatial patterns. Maybe it's niche formation, maybe it's not. I don't even know what it means. It's just that what you see, that's what you see. And loss of this negative feedback can result in loss of these sort of patched clumped structures. And again, we say that the hypothesis that comes out of this is that spatial interactions are crucial for cell lineage control, and we need to do more work now to build this up and to investigate this in a broader setting and put more realism and so on. But that's a first step. So far, I've talked about tissue homeostasis. Now I want to change things around a little bit and move to unbounded growth, tumors. And also we're going to travel back in time a little bit because this is stuff that we've done previously, but it's nevertheless related. Whoops. So we go back to the simple ODE model. And we forget about space right now. We've done it all in space as well, but it's easier to explain in terms of these models. So this was the initial model that we started out with. We had these two feedback mechanisms, one on the division rate, the other one on the probability of self-renewal, negative feedback. And I said that this one highlighted in yellow here is the one that contributes to stability. Okay, so if you want to have a tumor, that starts growing out of control, you got to lose that feedback on P, on the rate of self-renewal. So you can lose that feedback in two ways. You can either lose all feedback, and then you essentially have a population that doubles at defined time intervals. You get strong population growth, and that's here. You get essentially exponential growth. So the log number of cells in these simulations, you get exponential growth of the tumor cell population. There's no feedback left anymore, and we call this uninhibited growth. The other way in which you can lose homeostasis is you just lose the feedback on the self-renewal probability on P, but you still keep the feedback on the rate of cell division on R. And it's possible that tumors, at least in the early stages, still have some of the feedback loops that are present in the healthy tissues, because the tumor maintains a tissue hierarchy. So why not maintain some of the control mechanisms that exist? So if you have a situation where this feedback loop on R is still present, again, you get unbounded growth, but this unbounded growth is not exponential due to the presence of this feedback factor. Okay, and you get what we call inhibited growth, which is essentially sub-exponential growth here. We've done this in space with an agent-based model. The same results apply. If no feedback is there, you get uninhibited growth. With perfect mixing, it's exponential. In a 3D spatial model, we get cubic growth due to the spatial structure. If you've got the feedback, sorry, this is not supposed to say feedback on self-renewal, but feedback on division rate. If you keep that in the growing tumor, you get the inhibited growth type, which is sub-exponential for perfect mixing. And in the 3D spatial setting, it's subcubic. It's lower than cubic. We did some studies trying to look at tumor growth data that are out there of various spatial and non-spatial tumors. And you can fit those pretty well with mathematical models that assume either lack of feedback or the feedback still present. So there's quite a bit of cases where without any feedback, you don't get a good fit, but with feedback, you get a better fit. And so we think maybe that puts some, at least gives some confidence that maybe it's an indication that perhaps feedback factors determine these growth patterns to some extent. So an important point I want to come to is feedback and stem cell enrichment. What is the fraction of stem cells? So we've got these two growth modes. Without feedback loops, when you have uninhibited growth and straight exponential growth, you get no stem cell enrichment over time. The fraction of stem cells stays constant. This is the dashed line of the stem cells, solid line the differentiated cells. However, if you have the inhibited growth with the feedback on division still present, you get stem cell enrichment over time, where the stem cells eventually become the dominant population. So from that, one interesting aspect that emerges is maybe if these feedback loops are still present in the tumor, they may be contributing. They may be one of the factors that contribute to the enrichment of stem cells. There are obviously other factors that contribute as well, and many people have worked on it. Heiko has worked on that extensively, but that's one sort of mechanism. So why am I telling you this? Because stem cells are important for treatment response. So stem cells, if you go back here, if you've got a lot of stem cells here, stem cells, if you treat them with chemotherapy, they are much less affected by chemotherapy than differentiated cells. So they display a good degree of resistance against chemotherapy. So you can get stem cell-based resistance, and that can limit the impact of chemotherapy on the cancer. And I've been working with a guy called Keith Chan, who was at the time in Baylor. Now he's in Cedars-Sinai. He works in bladder cancer. He's working with the stem cells. He's working with patient-derived xenografts, and is interested in how does chemotherapy affect the cancer, and what are some correlates of resistance. And one thing that he found in those xenograft studies is that the cancer can become resistant without any obvious genetic basis in the cells, even if there's no resistance mutations where the drug becomes ineffective. In those experiments, he didn't find those mechanisms, but he did find that cancer stem cell enrichment occurred, and that this may be responsible for resistance. And so here, this is a bad slide if there are any clinicians around. Usually good enough for mathematicians. I'm just trying to explain what a patient-derived xenograft is. You take a tumor out of a tissue and you put it in the back of the mouse and grow it there. Obviously, that's not quite the bladder. That's something else, but close enough for mathematical purposes anyways. So this is the system that he works with. He takes bladder tumors from patients, put them in the back of the mouse, grows them, so the whole stem cell structure and stuff is kind of still there, and then he treats them with clinically relevant chemotherapy regimes, which is kind of 5 cycles. And this is the tumor volume over time in the mice. Initially, the tumor responds relatively well to the treatment, but in additional treatment cycles, the response is lost, and eventually it's not there at all. And again, there are no resistant mutations that have been found in those cases. So why is that? It's probably have something to do with stem cells. Interestingly, there is some heterogeneity if you look at different tumors that are taken from different patients and put into the mice. So here's an example in the first graph where the treatment response is eventually lost. Pretty much a very, very, very low treatment response. Here is another example where the response is maintained much better. So a significantly bigger percentage of the tumor is reduced even in the 5th treatment cycle. And here, this is only 2 cycles, so it's not very meaningful, but these are very big responses. And he says in some mice, even you can get rid of the tumor. So why do you have this heterogeneity? So first of all, what is the mechanism that results in the loss of the response? And what can potentially explain this heterogeneity? So this is Keith Chan's work. So he determined that there's an interesting positive feedback loop in place here. So in his system, he considers there are quiescent stem cells, there are cycling stem cells, and then there are transit amplifying, eventually differentiated cells. Now, when chemotherapy comes in and chemotherapy destroys the differentiated cells, those dying cells release wound healing signals, PGE2. And that wound healing signal results in what he calls stem cell repopulation. This wound healing response results in the activation of quiescent stem cell and in an increased division rate of those stem cells. And so he hypothesized that this wound healing response results in new stem cell recruitment, new stem cell repopulation, and therefore stem cell enrichment. So you have this interesting wound healing response that might be leading to that phenomenon. And here are some data from the mice. And interestingly, these are untreated controls, and these are treated with chemotherapy. The black dots show treatment with chemotherapy, and the treatment responses eventually goes down, and you get a growing tumor despite the chemotherapy cycles. But you can treat it with a drug, celecoxib, which prevents the wound healing response from taking place, and then the number of cells remains much lower. So this data shows that if you block that wound healing response that mobilize the stem cells, then you get a better treatment outcome. So this contributes to the hypothesis that this wound healing response contributes to stem cell enrichment.
Can I ask Dominik, before you go on, that's intriguing. So
yeah.
Wound healing is obviously a complex process. So what do you mean by blocks the wound healing response? I know the wound doesn't heal, but what specifically is being blocked?
What is being blocked is the stem cell, that the stem cells get out of quiescence and proliferate in response to PGE2.
So basically they're not seeing PGE2.
Yeah, they're not responding to that signal.
Okay.
Yeah. So it's, when I say wound healing response, it's just because it's sort of an analogy. When cells die, you want to replace them, right?
So it's not wound healing in a wound healing sense. It's more
right. Yes.
Returning to homeostasis is what you mean.
Correct. Yes. Yeah. He calls it a wound healing response. So I do too. But you're right. Yes. Okay. So this is the data that argues that this PGE2 induced mechanism, what he calls wound healing response, is required to see the stem cell enrichment and to get resistance. And so he was giving a talk at UCI, and I started talking to him. And so I suggested it might be interesting to test this hypothesis with mathematical models and see what you get. And so we started a collaboration, basically. And this is the model that we used. This, again, simplified model is an ODE, but also it sort of contains the components that he thinks are important: the quiescent cells, the cycling stem cells, transit amplifying cells, differentiated cells. You get the dying differentiated cells secreting the PGE2, which is the wound healing factor, and that then drives quiescent cells into proliferation and increases the proliferation of those stem cells. We don't have any of the negative feedback factors that I talked about before. We just have this positive feedback stem cell repopulation response in this model and nothing else, which is what he thinks is important. And so we now ask, how does that influence outcome? And this is how it influences outcome. In this model, here you've got the tumor size. The shades are the 5 treatment cycles in the model. We have chemotherapy without wound healing in green. We have chemotherapy plus the wound healing response in red. So this is just switching it on and off in the model, which we can do quite easily. Obviously, much easier than an experiment. And on the right, you see the same thing for the red line for the chemotherapy with the wound healing response, and we plot the stem cell population in black, and solid line and dashed line is the transit amplifying and differentiated cells. And you can see that during treatment, the differentiated cells and transit amplifying cells, which are killed by chemotherapy efficiently, they plunge to low levels. The stem cells are not really affected. They go up a little bit due to that repopulation response. And so during treatment, you get a strong enrichment of stem cells. But then you go off the treatment cycle and the system bounces right back to where it was, the same stem cell fractions. The transit amplifying and differentiated cells bounce back. Stem cells are here. The stem cell fraction is the same as before treatment. Next treatment cycle, again, stem cells become dominant. They increase a little bit more, but then you go off the treatment, bounces back to the same fraction of stem cells as before. So you don't really get stem cell enrichment in the system as a result of the wound healing response. That's maintained in the long term after treatment. And then when you look at what is the percentage of the tumor that is being killed in each treatment cycle, in the experimental data that goes down, remember? And in this model, where you have the wound healing response, which is red, it does nothing. It stays the same. The percent of tumor that is killed stays the same with each treatment cycle, whether you have wound healing response or not. So based on this model, we said maybe we can reject the hypothesis that this wound healing response alone is responsible for the loss of the treatment response with each successive treatment cycle. Maybe there has to be something else in there that can contribute to this. And so based on what I showed you before, we thought more. We think that the reason that the treatment response is not reduced with each treatment cycle is because there's no negative feedback in this system in the model that we have. And remember, without negative feedback in this model, you don't tend to see stem cell enrichment. On the other hand, if you put negative feedback on the division rate of stem cells in, you do have stem cell enrichment. So we amended the model, and in addition to that wound healing response, you put negative feedback. The differentiated cells are assumed to secrete factors that inhibit the division of transit amplifying cells and inhibit the division rate of stem cells. So when you do that, you get very different dynamics. You see, let's look at this here. This is stem cells versus more differentiated stem cells in black, solid line, versus more differentiated cells in dotted line. So initially, there are more differentiated cells than stem cells. As before, during treatment, stem cells remain and go up, but differentiated cells disappear. But then when you stop treatment, it doesn't bounce back to the previous fraction of stem cells. In fact, the enrichment that you caused during treatment persists to some extent. Here you have a little bit more stem cells than just a little bit less stem cells than differentiated cells. But after the next treatment cycles, you have more stem cells than more differentiated cells, and more and more. And this results in a larger number, an actual increase in tumors. So it damages, it's counterproductive in an important way. And if you now look at the fraction, or the percent of the tumor that is reduced with each treatment cycle, we see that it goes down. It is large fraction killed at the first cycle, less in the second cycle, and even less and less with subsequent cycles. And we compare, in red, we show the chemotherapy plus the wound healing, and in green, we show the chemotherapy alone with the wound healing response switched off. The negative feedback is present in all simulations. We can see that without the wound healing response, the decline in treatment response is less pronounced. With a wound healing response, the decline in treatment response is much more pronounced. So what we conclude from this analysis is that maybe this negative feedback on division is important to explain stem cell enrichment, is important to explain the loss of the treatment response, and the wound healing response alone cannot do it. But if you add the wound healing response to the negative feedback, then you get the most pronounced loss of treatment response, which is the red line shown here. So they somehow synergize and collaborate to contribute to this emergent resistance. So the upshot here is we reject the hypothesis that the wound healing response alone can lead to loss of treatment response, and we hypothesize that a combination of the wound healing response with the negative feedback on division results in the loss of response, and that this negative feedback is crucial here, is a crucial component. So can we test this at all? So I just want to finish it off, last few slides, whether we can get any indication of whether this is correct or not. And maybe we can get some indication from tumor growth in the absence of treatment. So if you take a model and you look at negative feedback in the absence, growth in the absence of treatment, if there's no negative feedback, you get more or less straight exponential growth. If you've got negative feedback, you've got this inhibited growth, and the stronger it is, the more inhibited the growth gets. So here's got this cell, the PDX xenografts. Some are sensitive to treatment and some are more resistant to treatment. And so we compare how do they grow in the absence of treatment. And if they are sensitive, they grow much more exponentially. If they're resistant, they grow much more like the inhibited growth pattern showed by the model. So this is not conclusive. The data are weak at this stage. We can do some statistics that argue that those xenografts that are resistant during treatment, when they grow without treatment, they follow a more inhibited pattern. And so this gives us enough confidence to say maybe it is the negative feedback that actually contributes to the resistance through stem cell enrichment in these cases. So maybe there's something to that theory. So the preliminary data support the model prediction that more resistant tumors have stronger negative feedback. And we're now searching for relevant feedback, negative feedback loops in bladder cancer. And perhaps this could be used to predict the response to chemotherapy and could allow us to work out better treatment methods to get around this stem cell, the enrichment, stem cell enrichment and the consequent resistance. I think that the insight that maybe these feedback factors are important to make the tumor resistant can explain some of the heterogeneity. Some tumors have less feedback presence, some have more feedback presence that can result in differences in stem cell enrichment, that can result in differences to treatment response. And maybe that can be exploited therapeutically. So that's where we are with this, and I'll leave it at that. I just want to acknowledge people I work with at UCI, and importantly, Keith Chan's lab at Cedars-Sinai. Thank you very much.
Thank you very much, Dominik. Excellent presentation, and that's definitely a topic that I think a lot of us here are very interested about. Simple models, stem cell dynamics, homeostasis, cancer. At this stage, if I expect people to have questions, so if you have any and you have kept yourself quiet until now, this is a good time. I know Sandy has questions, but he's been asking them, so I'll wait a bit.
I have questions, but I want to try and let other people ask if there's any others.
So I have a general question about homeostasis, which is that obviously a typical view that I've seen before and I have wrote about myself is that cancers result from the disruption of homeostasis. So you need to first understand how the tissue is behaving in normal conditions, and then figure out how you can disrupt and how cancer can get initiated. But another sort of perspective is like, if this is your view, is there anything you can do in order to restore the homeostasis? Is there anything about your models that would allow us to sort of figure out what kind of interventions would allow us to restore homeostasis, or once the horse is out of the barn, then there's really no way of putting it back in?
Right. Yeah, I haven't thought about it this way, because I always thought once you've lost these things, it's probably sort of a genetic loss, unless you return, unless you sort of change the mutation back to what it was before. Then that might be hard, but there may be things like plasticity there, where you can change that. So I haven't thought in detail about that. One thing one can do to exploit this sort of regulatory thing is to, if you've got stem cells that are growing out of control, you can try to make them differentiate out and lose their potency, so to say, that, and drive the tumor down and drive it to a more normal state. And that's probably one of the things that one can try. And
the other side of this is that homeostasis is just more than I guess that a lot of these models are of epithelial tissue. So my question to you is, given that there's more cells involved in maintaining homeostasis in most organs, fibroblasts, for instance, would be an example, but I think I can come with a lot more other cell types. You don't think they're particularly important in this process, or are you just starting and you're considering now a more complex ecology of the tissue?
No, I think they're absolutely important, and it's important to realize what I presented here was a very specific question, and the model is tailored to answer that particular question. The question was, can this particular feedback loop that has been postulated before to result in homeostasis, can that result in homeostasis in a spatial setting or not? And we answered that question. Obviously, there are other things that contribute to homeostasis, and so here we look at lineage feedback, where cells of the same lineage try to inhibit each other, but there are also other cells in the microenvironment that are secreting things that are contributing to tissue structure, to homeostasis, to everything else. We have not looked at that, because that's not what the question we're asking. If we want to go into more detail about what determines homeostasis, we have to put in a whole lot more. But I'm always, when I design those projects, I'm always more in favor of one question, try to answer that question, and then move on in complexity.
Fair enough. Sandy?
Okay, so I'm intrigued about the kind of intermittent treatment results that you had there, because of course, we're very interested in switching treatments on and off and treatment holidays. in a way to try and control the tumor burden, this whole adaptive therapy paradigm, for example. You didn't look into that, but if you thought about trying with your model, with the combination of mechanisms, what happens if you do that? If you do intermittent treatments? Like does
yes. Yes, we've thought about it. We've written about it. We've written about it in grant applications, but we haven't done it yet. Yeah. So once, I mean, it sort of depends on our goal at this stage is, if we follow up on this, is to identify in this bladder cancer system what other control mechanisms are operating in that tumor that shapes the response to treatment, that shapes what happens during treatment, and that shapes what happens in the off phases of treatment. Once we understand that, we can then try to design treatment protocols that exploit this knowledge to try to figure out how can we optimally space the on and off treatments in order to keep the tumor in the long term at a lower level. But that really, the next important step, it hinges on us understanding the identity of this feedback factor. So we're now, based on this modeling work, we've postulated that these negative feedbacks should probably exist in order to explain the data. That's our hypothesis. We need to confirm that experimentally. And that's the first step. Once we've done that, we go further.
So I agree with you on some level that it would be important to identify that mechanism and pin it down. However, you don't necessarily need to pin that down to try and hypothesize a strategy and try it, right? You can just assume it's there and it works, and then play it out, right?
Yeah, absolutely. Yeah, that can be done, and maybe we should be looking into that. Yes.
I mean, one of the things that I see specifically in that, and your sort of death feedback aspect there, right? You know, there was a lot of talk in the early 2000s about this sort of phoenix rising kind of idea about, you know, stem cells emerging from the death of other cells. They weren't stem before even. So, you know, I'm not sure if that model could capture that, but like, if you were in a more differentiated state, is there a way back? Is there any way back in your model?
Right. I mean, we have looked at model where there is de-differentiation. I think that was we've got one paper on it, and it was basically some simple evolutionary question we asked. We haven't asked any question in this particular setting here, but that would be interesting to put that in, because this plasticity obviously sort of happens, and it contributes to what's going on. So that's another thing. I mean, one thing that Keith Chan, my collaborator, has is he has this interesting system where you can you can label stem cells and differentiated cells, and they glow in different colors, and he's got a high throughput imaging system where he can then follow the fate of these cells over time, and you can really follow these things, and you can use that to quantify what processes are actually happening in those in these stem cells, and you will be able to see if a cell de-differentiates from a more differentiated cells and a less differentiated cell. You'll be able to see what proliferation and differentiating patterns happen, what the kinetics are. You can parameterize the whole thing. So eventually that's what we would like to do. That's where we're trying to get to with this, and that'll give you very specific information of what's going on in the system, and that can be exploited then obviously in these ways we just talked about.
Sounds like an ideal experimental system.
Yeah, it is.
So yeah, sorry, I have a question from Michael Ratz. So if I get this right, he's writing us from Plön, and he says, thanks for the cool talk. Do the stem cell patches that are driven by the negative feedback eventually merge to a single large patch?
Say again?
Yes. Do the stem cell patches that are driven by the negative feedback eventually merge to a single large patch?
No, they do not. That is the equilibrium state in the system. You get these patches. The bigger your system size, the more of these patches you have, and they stay.
Cool.
Dominik, this is Heiko. Got a question as well. Very nice talk. Thank you so much.
Thank you.
I was very interested to see the evolution of the different subpopulations during this intermittent treatment or during chemotherapy on and off. Are these results robust or dependent on what's the functional form of the feedback you program mathematically?
That's a hard question to answer. So on a simple level, if it's done by the Hill function. If we take different forms of it, formulate that in slightly different ways, it's still maintained. But what if we do it more generally? My feeling is that it doesn't depend on this in this system, but it needs to be worked out more. So we looked at stem cell enrichment and feedback in a more general term, where we don't have a particular function for the feedback inhibition, but have a general sort of axiomatic approach where they have certain properties, and we have a class of models with these properties, and as long as these properties are there, we found that it remains robust, that this kind of feedback can result in the enrichment of the stem cells. So based on that, I would expect that in the system that holds true as well, and these results are not necessarily specific to the particular formulations that we've used, but are broader.
Okay, so the results you have shown us was the negative feedback on R or on P?
On R, for the stem cell enrichment. If we're talking about the stem cell enrichment and the treatment, that's a negative feedback on R, because if you've got negative feedback on P, that blocks unbounded growth. So you need to get rid of that in order to get unbounded growth.
But if you were to give treatment, then this block would be gone. You would get similar unbounded growth during the treatment phase, but then a bit more inhibition in the off-treatment phase, right?
What do you mean? I didn't understand the question.
So you would get probably similar evolutionary dynamics during treatment, that during the treatment you would release the self-renewal inhibition, and you would get enrichment of stem cells during treatment, but you would control it in the off-treatment phase.
I think so, yes.
Okay, very cool. Thank you.
Thank you.
Cool. All right, thanks, Dominik, for a great presentation and for answering all our questions.
Sure, it was nice to see everybody and to
yes,
my work.
Yeah, it's lovely to see you again, Dominik. Lovely work. Keep it up.
Thank you. Hopefully we'll see each other in person soon.
That's correct.
Okay. All right. See you later, guys. Bye.